My name is Laszlo Farkas. I'm an associate professor in the Department of Internal Medicine, Division of Pulmonary, Critical Care and Sleep Medicine, and the Davis Heart and Lung Research Institute at The Ohio State University. We are mostly focused on understanding pulmonary arterial hypertension. This is a severe chronic disease that is characterized by the progressive narrowing and structural change of blood vessels in the lung and increased blood pressure in the circulation of the lung. This leads to a severe impairment of function and shortness of breath in patients and premature death. And we are trying to understand one of the potential root causes, which is dysfunction of the endothelial cell. Currently, we have three main clinical treatment pillars that target different pathways, but what they have all in common is that they're mostly changing the constriction of the blood vessels, but they do not change the function of the endothelial cells fundamentally, or the process that leads to the structural changes in the blood vessels. So we're trying to overcome this by going exactly for these targets that can help to reverse the dysfunction of the endothelium and can change the structural remodeling in the blood vessels. We were trying to, we were looking at a mechanism of endothelial dysfunction, and we found, interestingly, that a molecule, it was called RAB7, this is a GTPase that you find in the endosome and lysosome of the cell. So the endosomes and lysosomes of the cell are part of a system that's called autophagy that regulates basically the removal of molecules and cell organelles that are not functioning. This process is called autophagy. You can see it basically as the garbage bin of the cell. In a way, we found that this molecule was reduced in the endothelial cells specifically from the patients. So we started looking at the potential implications for endothelial cell function in therapy. The main discoveries were, first of all, that we found that this molecule, RAB7, was specifically reduced in endothelial cells from patients with pulmonary arterial hypertension. Then we were interested in its relevance to the physiologic response of the vascular system, and we generated a mouse model with an endothelial-specific reduction in RAB7 expression, in the expression of this molecule. And we found that this mouse model induced spontaneously pulmonary hypertension and structural changes in the blood vessels, and also aggravated these changes in a mouse model of pulmonary hypertension. From there, we were interested in why this molecule plays such an important role potentially in the pulmonary vasculature. We used genetic manipulation of cultured endothelial cells to identify the different parts of the system that were affected. And we found, first of all, that reducing the amount of RAB7 in endothelial cells induced changes in the trash can system of the cell by basically inhibiting the degradation with accumulation of molecules in the cell that were supposed to be removed. So this particular research shows a new potential target for treatment of pulmonary arterial hypertension based on findings that we initially had in human patient samples. And we are focusing here on a specific area, which is a function of the endothelium, the cells that basically line the blood vessels from the inside. And we try to restore the physiologic function by manipulating the cells. And this molecule, RAB7, in endothelial cells has a fundamental role, as we identified by the fact that removing it from endothelial cells in a mouse model induces spontaneously pulmonary vascular disease. We believe that because of the complexity of the connected downstream pathways, including the endosome, lysosome, or the trash can recycling system in the body, the mitochondria, the general cell function and the role in accelerated aging of the cells, that this is a valid target which we can use to restore many of the cell functions. That's why we are so excited to find this activator that can help us to accomplish this goal.